So hello and welcome back. In the last short video, I introduced to you the general concepts of metabolomics, of comparative metabolomics, and the ideas what metabolomics can do for us. What I want to do now is to introduce the field of environmental metabolomics and especially the investigation of plankton communities. Basically, if you look at our globe, the entire globe is covered to more than 70% with water. And in that water, life is really happening all the time. In fact, the primary productivity, which means the global amount of photosynthesis and fixation of carbon in the plankton, is roughly equal to the contribution of all land plants taken together. So basically, if you look in the ocean, what you see is really a very complex community that is fueled in the energy by sunlight. The sun provides the light for photosynthesis, and this means that in the ocean there are, for example, small algae. They are microscopically small, called the so-called phytoplankton, and these small algae, they fix the carbon, they divide, and they fuel the rest of the marine food chain. So basically, those algae, they can be taken up by, for example, herbivores, small, for example, copepods, that are then feeding the higher trophic levels like fish. So this is really what is the base of the marine food web. But also, these algae can interact with other organisms, like, for example, bacteria, or with viruses. And together, they make up the microbial community of the plankton. So given the enormous importance of that system in terms of global CO2 turnover and in terms of global productivity, we are very much interested to learn about the functioning of this entire microbial community. And we do so by going out into the ocean and taking a research vessel, the Tara Ocean, and with this research vessel we can sample the marine life. We are on board and can generate subsamples of the marine life, including basically all the organisms that live there at a given time, but also all the water that they are living in. And now our general hypothesis is that these organisms, like for example the microalgae, they grow under the limitation of the external energy input. But on the other hand, they can interact with all the other microorganisms within the plankton, and they can do so by means of chemical signals. In fact, such a microbial community like microalgae, bacteria, and viruses, they constantly communicate. They ask who is there, they ask about the fitness, they ask if they can be acting as pathogens or as supporting symbionts, and all that can be unraveled if we understand the chemical signals that mediate these interactions. So what we can do now, if we have such a sample in the lab, is we can do a sequential filtration approach, which means we take now our water that we collected from the sea and filter it through the mesh to at first pick out the really big organisms. The water with the smaller organisms is flowing through and can then be administered to the next finer sieve that, for example, holds back then the microalgae. And this water can be taken and can be again filtered through a very finely porous sieve, which is holding back, for example, the bacteria. So we have now 4 different fractions. 1, the large organisms, 2, the smaller ones, 3, the microorganisms, and very importantly, 4, the dissolved organic material. And now what we can do is we can take these samples and use them for metabolomics. So we take our sailboat and we sail along the coastal lines of, for example, the South American continent, as we did with the Tara Oceans. And we can sample along that coastal line where the rivers flow in or where you have upwelling water in order to see, in the first place, with genomic methods, the inventory of species that are there, but also the compounds that they produce. Now, what we can do is, if we take, for example, the sample number 2, which are the algae themselves, we can ask those algae, how do you perform? We look at the compounds that are found inside the cells and that reflect the fitness and the performance of these cells. But we can also look at the compounds that are released out of the cells that reflect basically the chemical language that they can speak with the organisms that they interact with. So basically what we do now is we take those samples that we generated from the sea, like a snapshot at any given sampling station, and profile all the compounds. So we can, for example, ask what is a cell releasing at a given time. So we sample the water number 4, and we generate, as I explained in the last video, so-called chromatograms, intensity versus time, that reflect basically all the compounds that we find in the seawater. And we can, again, using metabolomics techniques, ask for each and every compound, who are you? What is your chemical nature, by looking at the mass spectrum. So by doing so, and by going throughout the coastal lines, we can, in fact, generate now chemical maps of the inventory, what the cells release into the ocean. And if we ask then the biologists on board who do the genetic survey or the ecological surveys, what do you observe as interaction partners within the plankton community? And we can correlate the chemistry that we record there. We can learn about the chemical language that is spoken by the plankton, and we can learn how such a complex system with hundreds and thousands of microorganisms in a given liter of seawater is organized and is regulated. By doing so, we can, for example, now identify defensive compounds that are mediating the chemical defense against higher grazers. We can look at bacterial resistance by the algae. So if we see a co-occurrence of pathogenic bacteria and microalgae, we can always ask, how does the algae respond to the bacteria or to viral infection? And if we do so, we can assemble now a global map of the chemical landscape, a global map of the organismic interactions, and if we bring them together, we learn about the functioning of the plankton. Thank you very much for the attention.